So first you take blood from the individual, you centrifuge this blood. You do this in the lab using special machinery. So after centrifugation, the blood will separate into red blood cells, white blood cells and platelets, and plasma. You are going to take the white blood cells from the middle layer and culture them at 37 degrees Celsius for 3 days. When we say culture, we mean you place them in glassware that contains a medium where they can grow and live. So it's going to have oxygen for them, it's going to have nutrients, and so on. The temperature is 37 degrees Celsius, the same as the body. You leave them for 3 days so that they can multiply and grow, so we can have a lot of them. After that, you add colchicine and hypotonic saline. These will cause metaphase blocking, so the chromosomes will be stuck at metaphase, which is the stage where they are the most condensed and the clearest, so that we can see them and place them in a karyotype. While hypotonic saline is going to cause the cells to burst in order to release the chromosomes from the nucleus and from the cell. So colchicine will cause the chromosomes to stop at metaphase, and hypotonic saline will cause the white blood cells to burst so that we can release these chromosomes that are stuck in metaphase. So now in the cell culture, we have chromosomes stuck at metaphase that are released from the cells. We take them and we fix them onto a slide, and then we spread them on this slide. After that, we stain them so that they can be colored and we can see them more clearly, and also so that we can identify them, because they will be stained in different patterns. Each one has a specific pattern for the kind of chromosome that it is. So because of the staining patterns, we can tell this is chromosome 1, this is chromosome 5, and so on. You analyze them, photograph them, and you cut and paste the picture of every single one of them, and you order them. As I said, you can tell which chromosome is which because of the pattern of staining that it takes. And you paste them and arrange them together in homologous pairs depending on different criteria, like the size, the staining patterns, and so on. And this is how you obtain the karyotype. You arrange them in homologous pairs depending on the size, the position of the centromere, if it is towards the upper part, lower part, if it's exactly on the middle. There's references that you use in order to compare, to know which chromosome you are talking about and which ones are paired together. And also we have the dark and white banding patterns. If you can see here, the chromosomes are stained with different banding patterns. And we use references to know from the banding patterns which chromosome this is. So this helps you create the karyotype. And that is how we created karyotypes from obtaining blood samples. We did this for a human male and a human female, and that's how we obtained the karyotypes that we saw earlier. And this is it. This is karyotyping.